Good afternoon, everyone, and welcome back to the 26th Wisconsin Symposium on Emotion. We have a really fantastic afternoon planned for all of you, and it's really my honor to welcome our next speaker, Tracy Bale. Tracy is a professor of pharmacology at the University of Maryland School of Medicine, where she also directs the Center for Epigenetic Research in Child Health and Brain Development. She has published hundreds of papers and is an extraordinarily accomplished neuroscientist. She's won numerous awards, and we're not giving extensive biographies. If you wish to learn more about any of the speakers, please go to the Health Emotions website, and you can also find the articles that each of the speakers has provided as the basis for their presentation. So it's really fantastic to have you here today, Tracy, and I welcome you to our symposium.
Thank you so much, Richie, and I want to say thank you both to you and Ned for the invitation and the opportunity to participate. I think this is something we've discussed over several different years, and I'm glad, well, I'm super happy to be here today. I really wish we were in person because I know we all miss that personal interaction so much, especially the opportunity to talk about science. So today I want to shift gears a little bit, not in what the focus of my lab's research is in terms of the other talks you've heard today, but to really start way back in time and how we end up with the brain. And as Richie said, my lab and my research interests focus on stress and understanding the role of stress and how we respond to our environment in terms of stress, but also all of the contributing factors. And I think if you were able to listen to David's talk earlier today, this is very much plays off of what David's message was in terms of all the contributing factors giving rise to disease. And I'm sure all of you are well aware, looking at our current pandemic and the increase in mental health concerns, especially related to anxiety and depression, as well as other neuropsychiatric diseases, that they are very much on the rise and contributed to by this incredible pandemic and the stress that we're all under. And you can see from these statistics put up by the CDC that they're still increasing. Who is at most risk? You can see here, women are showing higher rates of mental health concerns during COVID, especially those women, if you see on the right side here, who have children under the age of 18, is a contributing factor and an additional stress to worry about and deal with during the pandemic. But if we also look by race, it will not surprise you to find out that the burden of the risk factor for anxiety, depression, or other concerns, especially brought upon by the pandemic, are exceptionally high for people in our Black communities, with the addition, of course, of racial discrimination and health disparities that they unduly share the burden of. And so with that, I think you've heard this message throughout the day, and I have no doubt that Josh will talk about this also later, which is when we think about any neuropsychiatric disease, but especially those of neurodevelopmental origin, defining who is at risk. So whether you're studying a clinical population, developing a new treatment, or looking at an animal model, defining the risk factors to try and understand ultimately where to intervene is very complicated. But I think thinking about this as a gene by environment by developmental window really gets at the heart of all the aspects that we're all concerned with. And I want to highlight that gene can be meaning of many things, everything from SNPs to the types of genes that are on X and Y chromosomes. And I highlight down below here that sex is a biological variable, very much intersects with all of these components. So as I said, X and Y linked genes we now know are critically important for brain development and as well as disease risk. The environment in which you can look in the preconception environment during pregnancy or all stages across the lifespan, including those effects of stress during those windows, but also the developmental windows themselves. And I want to highlight, because I will come back to this multiple times in thinking about what those developmental windows mean. It is not just the gestational period in which pregnancy and perturbations of mom, but it's the events that mom and dad bring to conception, as well as the rates from those signals of mom and dad the rate of brain development during gestation, early postnatal development, that can also be key factors in understanding who's at risk. So as I said, my lab is very interested in stress and disease risk, but one of the things that we all need to think about is what are the contributing factors starting at the point of conception, coming from both mom and dad, that give rise to who is at risk and what those factors are so we can better study them. And you've heard already today some great talks on all of these aspects, all the way down to the level of individual neurons. So I'm going to take you a little out of your brain comfort zone and go back to things like sperm and egg and talk a little bit about the reproductive processes and which underlie many of these disease risk components that we need to understand better. So to understand during pregnancy where brain development is happening for the baby, we also need to understand the components being delivered at conception by both the egg and sperm, and therefore by mom and dad's preconception environment and those signals they bring together. And these are not likely to be those individual genes we may be used to thinking about in terms of the brain, but rather they are signaling components that give rise to things like speeding up the course of development, slowing down the course of development, in which for the brain itself may allow the brain to be at greater risk, missing either selective pruning area or times of development and or speeding up the rate of delivery at which the species survives, which evolutionarily makes sense, but things like the refinement of the brain may be a consequence. So we think about also the components that mom brings to the period of pregnancy, of course, and those can be biological in nature, such as those components of her uterus. We also in my lab think a lot about this other tissue here called the placenta and the development of this very dynamic critical tissue as it gives rise to transplacental signals that tell the developing baby everything that it needs to know about the environment it's about to be born into. One of the key things I want to talk to you today about, and this may be familiar to many of you, are extracellular vesicles as a potential really innovative biomarker that allows us to do translational research between animal studies and humans. So for those who are unaware, extracellular vesicles are a great model in which we can understand how signals traveling in circulation at high concentrations between tissues, largely in signal to the immune system, have largely been studied a lot by cancer biologists. But neuroscientists are now beginning to understand and appreciate the value of extracellular vesicles. These travel, as I said, in circulation in all mammals. They're secreted by all tissues, and they travel in the body at high concentrations. I'll talk a little bit about these vesicles as it relates to our animal studies and our translational human studies as well. Key components of these nanoparticles as they travel in circulation are that they have high concentrations of proteins in their lipid membrane, and the inter-nanoparticle components are largely small non-coding RNAs that act at the distant sites of which the proteins determine their specific action. And you can see here on the right, over the course of time of discovery of these extracellular vesicles, we've learned a great deal, both about the size and different types of extracellular vesicles, which includes, probably many of you refer to them as exosomes. Exosomes are just one small type of extracellular vesicle. These vesicles are produced intracellularly from different components and compartments and are secreted in many different ways from individual cells into circulation. And as you can see, we now think of them in many different ways based upon their size and the components and the tissues they're secreted from. So we'll come back to the extracellular vesicles. In thinking about race as a component of an additional stressor that Black communities and other individuals of color endure on a regular basis, and the accumulation of this is called weathering. This has a systemic effect and contributes greatly to the toll stress has on the body and to neuropsychiatric disease risk. So our lab, in collaboration with Tanya Jovanovic and the Grady Trauma Project, recently set out to determine if we could isolate extracellular vesicles and determine specific and unique components of those vesicles, giving rise to indications of when particular trauma may have happened across the lifespan. So in order to ask about a timing sensitivity or timing specificity of a particular type of trauma, interpersonal violence, in this case, these are from women who've experienced sexual trauma. We used this particular trauma to assay because we knew that it was a less fraught with error in terms of timing of when it was endured and could be more specifically identified as the timing during either childhood or adulthood when it was experienced. So you'll see then using trauma questionnaire in the Grady Trauma Project that Tanya was able to isolate These are now women in their 30s into their early 40s. These are either experienced sexual trauma or interpersonal violence prior to adolescence, during adolescence, or only in adulthood. These are women from the inner city of Atlanta who have an overall high level of trauma experienced and a lot of racial discrimination. This cohort is about 97% Black. We looked at many different physiological measures in these women now that they're adults. We did a liquid biopsy, and the only time I have today to tell you about is focusing on these extracellular vesicles. But we've looked at other components as well, including cell-free mitochondrial DNA and circulating cytokines. So this just tells you a little bit of the data we were able to obtain on the physiological outcomes. On the left here showing you the body weight of these women, remembering again that these women are in their mid to late 30s. There is about 25 to 28 women in each category here across. You'll see that we're first separating them out, doing statistical comparisons between no sexual trauma versus sexual trauma altogether. And you can see that, and this has been reported previously by others, that this type of interpersonal violence leads to a lifetime accumulation, demonstrating increased body weights, body mass index, increased waist circumference, and hip circumference as well. And you can see when we parse this out by when that trauma happened, you start to see an effect that looks like it's accumulating around for women who experience childhood interpersonal violence. If you then take that data and look at it in terms of a risk for hypertension, again, we start to see a higher level of hypertension in the women who experience sexual trauma. And what's really interesting, if you parse this by what the CDC frames as hypertension stages, you can see that the women who experience interpersonal violence in childhood, and then in adolescence, and then in adulthood, you start to see an earlier onset of hypertension stage 2 risk compared to women who have had no sexual trauma. Remembering again that all of these women have a paired level of overall high trauma in the community. When Tanya began looking at these women for looking at things like PTSD and fear-potentiated startle, you can also start to see, parsing out in this data set now, some specificity of interpersonal violence, especially if you look at the fear-potentiated startle for women who experienced IPV during adolescence specifically. And we're now looking further into this data. But what I wanted to show you related to the biomarkers that we're beginning to associate with this physiological readout and looking at the extracellular vesicles that I told you about earlier. So we isolated extracellular vesicles from these plasma samples. They were all obtained during the same period of time, shipped to us, and all of the samples isolated for EVs at the same time in the lab. So we're first comparing for overall by proteomics. So this is looking at the protein levels in these nanoparticles. First looking by no sexual trauma compared to sexual trauma for these women. And you can see there are clear distinctions in specific proteins. If we then parse them out by when the sexual trauma happened for this group here, you can see on the lower end here, these pink are lowly expressed proteins here, which are identified here across all sexual trauma groups. Those are largely immune and immunoglobulin related proteins. The other thing I want to highlight here is that this specifically in the women who experienced sexual trauma just during adolescence, these blue colors here, which on the Z-score you can see are enormously increased proteins, turned out to be some really interesting differences in all keratin related proteins I'll come back to in a minute. I wanted to show you overall here, looking at log 2 fold to change. So these are really big differences overall. These are not mild effects that we see in key components that tell you a lot about extracellular vesicle secretion and how these extracellular vesicles are being released to travel in circulation, that annexins and galectins, which are key proteins for extracellular vesicle function, are significantly and robustly reduced in this population of EVs, specifically in the sexual trauma experienced women, which may tell us something about hallmarks of how the stress programming has occurred across the women's lifespan. But what I want to focus on is getting everyone who works on stress and neuropsychiatric disease to think a little bit different from this data set that we've uncovered here. So as stress researchers, we think a lot about the autonomic nervous system, the HPA axis, but one of the organs that we oftentimes do not think about is the skin. So this data set brought to mind for us because of all these keratin-related proteins that you see in this bubble plot here that were dramatically increased. In fact, many of these proteins were only present in the women who had experienced this trauma during adolescence and in no other group, which is really interesting. But the skin is the biggest barrier, and it's the largest organ responding to stress, and no doubt is a very big component of how we react to our environment, both at the level of the skin and the hair follicle. So what this might mean, and whether it's unique to women or women of color, we don't know. We're now comparing those studies in larger groups. Is it interesting to think about as a biomarker for who may be at the greatest risk for things like PTSD. In further investigation of this data set, as we began to explore this novel area of how these keratin-related proteins, so these are both keratin-associated proteins and keratin type 1 and 2 type proteins, what came to light was that the majority of these proteins only in this one group of women were actually part of a 17q21 loci. So they're linked to a very specific region. This is super interesting to try and figure out if there is a potential epigenetic mechanism that occurred during adolescent trauma that programmed these unique proteins as part of a systemic signal related to stress. It also of note is very interesting that in this 17q21 region are also a number of associated links with risk for asthma and several other stress-related disorders and immune function. So it's something we're currently exploring. And again, to determine if this is just unique to women or just women of color, I think is something critically important to understand for disease risk. So as I said, the accumulation of these stressors across the lifespan, especially during periods of incredible stress demand, such as pregnancy. This is Arlene Geronimus here, who coined the term weathering, and noting that the stressors that impact women and women of color are chronic and repeated through the life course, and that discrimination stress is another feature of which impacts, especially as it relates to preconception stress itself as a component. So I say preconception stress, many of you may think of this as thinking about ACEs, or adverse childhood events experiences that we know from the Kaiser Permanente that outline the fact that the accumulation of these ACEs in childhood determines a lot of risk for many disease states across the lifespan. But the accumulation of 4 or more seems to be the point at which you are at the highest risk for many diseases, including those of mental health, behavioral outcomes such as addiction, and that those components can also give rise to further increased risk for neuropsychiatric disease. So at the period of pregnancy, understanding the contribution of ACEs or preconception stressors, if we look at pregnant women, so women currently pregnant across the United States, that key point of 4 or more ACEs from their childhood, you'll note that about 12.5% of women across the United States while they're pregnant have experienced 4 or more ACEs. However, if you look within the city of Baltimore, and not surprisingly, you'll note that 61% of women in the city of Baltimore who currently deliver at the University of Maryland, so a majority Black city and a majority Black population, have experienced 4 or more ACEs. So I want to highlight here that it's really critical when understanding the population in which you are, of which we are serving here at the city of Baltimore. I think it's really important to understand the community in which they live and the community that we are serving, as there are great consequences for this at-risk population. As you can see here, that in the United States of America, still in this day and age, that Black women, regardless of socioeconomic status or education, remain 3 to 4 times more likely to die during pregnancy, as do their children as well within the first year. So the mortality rates for both women and their children, for Black women and children in pregnancy and the first year postpartum, are 3 to 4 times that of white women, regardless of socioeconomic or education. So how do we think about extracellular vesicles and defining who and which of these women could be at the greatest risk? And could we find ways to intervene and identify new strategies for what that intervention might be? So again, thinking about these extracellular vesicles, one of the key components we need to understand about extracellular vesicles traveling in circulation is that they're at very high concentrations. But the point in the lifespan at which they are the highest is during pregnancy. And that is largely because the placenta tissue secretes tons of extracellular vesicles into mom's circulation. So Joel Sadowski at the McGee is the leader in this field and has been working in this area for decades. And to understand the different cell types within the placenta, the secretion of these important stress signals into circulation, again, largely to signal to mom's immune system. But Joel has done a ton of work to demonstrate that these extracellular vesicles serve as an autocrine signal within the placenta itself. And we think that they may be a signal in which they can sort of coordinate actions of the placenta to coordinate activity between the maternal milieu, the developing fetal compartment, and ways to understand and appreciate the stressful environment that mom is currently living in. So again, there's many different components of these extracellular vesicles that can be examined. So how do we take what we know in the human and model it in the mouse? As you've heard today, that animal models are vitally important for understanding of neuropsychiatric disease risk and development, but the cellular differences between, obviously, and the brain differences between mouse and human are large. But we've noted that one of the things that makes the cellular level studies more translatable is to appreciate that post-conception, that once the embryo begins to develop, post that zygotic stage, remember that this green area of the developing embryo here is the inner cell mass that will give rise to the developing fetus. These purple cells on the outside here of the embryo are giving rise to the placenta. This is the trophectoderm, so these will become the trophoblast cells that invade and become part of the maternal decidua as well, giving rise to that final placenta. So you can imagine if mom and dad bring different signals at the point of conception, you could already end up with very different outcomes. Again, the rate of that embryo development, the signals that give rise to differences in the trophectoderm that may determine transplacental signals throughout the course of pregnancy, all of these signals are critically important for the ultimate brain that we're all very interested in understanding. So this is just a summary of some of the studies over the last 2 decades in the lab. Thinking about the maternal milieu, my lab has done things like maternal stress and dietary challenges. Maternal immune activation is also something critically important to understand. So understanding this F0 maternal compartment, because we're all interested ultimately in this neurodevelopment going on here in the fetal compartment, as well as, of course, those germ cells that will give rise to the next generation. But there's also this placental contribution here. This is not a sponge. It does not just wick information. It is actually an endocrine tissue, and it is sex determined by the developing fetus. So whatever the sex of the fetus is gives rise to the sex of the placenta. We know that neurodevelopmental disorders are at much higher rates in males than females, and there's a lot to be discovered in terms of the sex specificity and the role the placenta plays. I don't have time to go into all of our prenatal data, but we're trying very hard right now in collaboration with Alex Bonin at USC to try and understand some of these transplacental signals that we can then take as a translational study back into the human. So what is the signals that are changed? What are the signals that are changed, whether it's a dietary challenge or immune challenge in mom's compartment here, that give rise to differences, whether it's proteins, steroid hormones, that give cues to this developing brain to develop at different rates. Everything from the rate of the cortical, how the cortical neurons might be developing, as you heard earlier in talks today, to how this fetal rate of development might be changed from cues, as well as these extracellular vesicles that are being secreted at high levels from this placenta. This is just some data from the lab that we've accumulated looking at mice and understanding differences in both the small non-coding RNA and this heat map on the left here in pregnancy and pregnancies with stress and what those signals might mean for mom's immune system. And then on the right is a proteomics assessment of these EVs traveling in circulation. And as you can see, with stress shown here in red, a robust reduction in those immune system signals coming from the placenta in circulation. So just with mild stress experienced by mom in pregnancy, we're already starting to see these big differences. So going back again to the contributions of what mom and dad bring. So this preconception environment being so critical and thinking about the egg and the sperm. They both bring unique signals. It is not just mom and dad's DNA. There are many components here. While one sperm, for instance, may fertilize this egg, think about the millions of sperm that are showing up at the egg, all of them bringing extracellular vesicles from traveling the reproductive circuit to get to the egg. What do those signals do, and how do they determine differences in the rate of development that might be important for how that baby and then ultimately that adult thrives in their environment? So we often think about mom, but what we don't think as much about is dad. And these are studies that have been funded by the National Institute of Mental Health over the last decade, and thinking about the contributions that dad brings to the picture as well for how the baby's brain might develop. And we know from tons of epidemiological evidence, whether it's the Dutch hunger winter, the Swedish famine studies, and other studies that have shown that there is a role for dad's environment long after he's gone through puberty, for those changes in dad's sperm content to give rise to differences of signals at fertilization. And we've done studies on this, as have many others now, looking at the components of stress and how you can see in many publications there on the bottom. This model that we've developed in a mouse to try and begin to understand what are the translational signals we can explore in the human. So in this mouse model of male mice being exposed to what ultimately is 4 weeks of a chronic stress, and then we ultimately can breed those animals for any period of time after that stress is over, and they will continue to produce changes in their sperm that give rise to male and female offspring, repeatedly breeding them with a number of females. As long as we've bred them out after the stress is over, we continue to produce both male and female offspring with significant differences in their neurodevelopment, where those offspring as adults are hypo-stress reactive. And I'll show you that data in a second. We've done an exhaustive analysis of the small non-coding RNA composition of the sperm. We know it gives rise at the zygote level already in differences in rates of development and long-term changes in the offspring brain. This is the HPA axis outcome of the offspring from this mouse model. This is all old published data. This is showing you male and female offspring. This is an HPA stress response in these mice. These mice the males were bred 3 months after the stress was over. The dads had either experienced chronic stress or what we call double chronic stress. We wanted to know if we increased, was there a dose-responsive relationship of dad's stress? There's not. His offspring, male and female, are hypo-stress reactive as adults. What's interesting in understanding the cellular process, as I'll show you in a minute, is that if we breed these males right either during the stress experience here of 4 weeks of stress or right at the end of the stress, they don't produce a phenotype in their offspring. There is some component, and I'm going to give a shout out to Bruce McEwen and all of his career work on allostasis that I'll come back to in a second. And that is once the stress has resolved in this animal, then the changes become what seemingly are permanent, but not immediately. So that's something cellular signal that we're working on. This is showing you the sperm changes in the microRNA composition. There are hundreds and hundreds of microRNA in all mammal sperm. This is showing you very few changes between control and stress dads 1 week after stress, but really big changes in their sperm composition 3 months after stress has ended. Why? How does this happen? So sperm are transcriptionally inert, meaning going through the course of spermatogenesis, they're not going to respond to the environment, and they are transcriptionally inert once they enter the epididymis. So how is it possible that they're responding? How are those small non-coding RNAs that I mentioned changing? How are these epigenetic signals found in sperm changing if the sperm are transcriptionally inert? This has been an ongoing debate in the field, and only within the last several years has information really come to bear. I'll share with you in a minute. The ultimate question I think we all need to ask, especially in the field of psychiatry, is the why, right? We can all find differences at different levels, but what do they mean and why would they happen? So I wanted to highlight for a second, for those of you who don't spend tons of time thinking about reproductive processes, that the why seems to be because in this course of spermatogenesis happening in the testes, there's a blood-testis barrier. And the reason evolutionary for that barrier is to protect dad's DNA from anything happening in the environment. So that makes sense. But evolutionarily, how would dad otherwise respond to the environment? So there's all of these processes that occur over the course of spermatogenesis epigenetically to protect dad's DNA. There's a lot of MeWe and PeWe proteins that are really critical to protect from transposons and from the environment while in the testes. But once dad's sperm leave the testes, it enters in this caput or head of the epididymis region. I'm going to show you this quickly here. Once the spermatogenesis period is over, it is pushed by fluid motion into this caput region of the epididymis. At this time point, it is required for maturation. The sperm are not mature. They cannot swim at this point. They require, in a static period of weeks, for all mammals, the sperm hang out in these tubules, and they're receiving critical information from these epididymal epithelial cells here. And as you can see, this includes all kinds of growth factors and a large amount of extracellular vesicles. So now you can begin to imagine signals from dad's environment that have occurred over a lifetime can be stored in these somatic cells of the reproductive tract, being released to these sperm and ultimately carried to the egg and transmitted at the point of fertilization in terms of rates of development. So a brilliant graduate student who's now a postdoc with Ian Mays at Mount Sinai did this great causal study, because I think it was David Lewis earlier in his talk that highlighted, again, the importance of the 5 C's, one of which is causal. And I think that's a really critical point for us all to consider when we find differences, is what do those differences mean? So Jennifer Chan was able to grow up in a dish here, modeling stress in a dish, because it is impossible to collect epididymal epithelial cell, these little extracellular vesicles here in these tubules of a mouse, not possible. So she modeled it in a dish. So these are DC2 caput mouse epididymal epithelial cells. And she was able then to collect secretion into the cellular medium here, the extracellular vesicles. And what I'm showing you in all of this data that I'm not going to go through today is basically her thesis work as a pharmacology graduate student at the University of Pennsylvania when we were there. This rank studies here and these other proteomic studies on the right here, I'm just going to summarize for you by saying this was the way that she validated how she could develop an in vitro model of an in vivo outcome by demonstrating that the changes in the extracellular vesicle components in vitro modeled those of the in vivo sperm. Okay, so it took a long time. She was able to do that both at the level of the changes in the small non-coding RNA, as well as the protein composition here. And you can clearly see from this beautiful heat map on the right that if she compared the extracellular vesicles long after the CORT or the stress hormone treatment occurred in the dish, these extracellular vesicles remain dramatically different and very similar to the sperm content we see in vivo. These are just some of the other composition of the EVs. I also wanted to highlight for those interested in the glucocorticoid receptor that we've been able to identify this allostatic somatic programming, ultimately is what we're calling it, and that both in vivo using ribotag assessment, that we know GR, not surprisingly, is a key component. We can compare both genetically and with stress in vivo, these over 2,000 genes that are giving rise to changes that we see in large part responding to chromatin regulation. So Nicole Kanyuk is an MD PhD student that has now completed some beautiful cut and run assay, just demonstrating that the H3K27 trimethylation mark in these somatic cells mirrors in vitro what we're seeing in vivo. So we know GR seems to be the orchestrator. No one's going to be surprised by that in allostatic state. I think this is really interesting based on some of the talks we've heard today, that while this is an epididymal epithelial cell, no surprise that you're seeing the same dysfunction around chromatin remodeling, and we see mitochondrial changes similar to what David was showing earlier in the brain. So I'm going to leave you with this beautiful work that Jen was able to do towards causality here using ICSI, or intracytoplasmic sperm injection. This is a beautiful study she put together by taking EVs, either from vehicle-treated or previously CORT-treated cells in culture, taking one dad, so dividing his sperm into 2 pools, incubating the pools with either the EVs from the vehicle or previous CORT treatment, washing that sperm, taking eggs from one mom and dividing them into 2 pools and injecting by ICSI the sperm from these 2. So now you have sperm from the same dad, eggs from the same mom, the only difference being the pool of EVs they had been incubated with, injected into or transferred into a surrogate mom on the right and left side of her uterus. So now we've controlled for all the confounding factors. Another C that you heard from David earlier today of the 5 C's. So same sperm, same egg, same intrauterine environment, the only difference being the EVs. And then we're able to do a number of studies where Jen could sacrifice the animals at mid-gestation and ask about brain development, as well as look phenotypically at the offspring. This was the ultimate end to her thesis, which was beautiful work showing you by bulk RNA sequencing that in fact there's dramatic differences already at mid-gestation in their brain development, and ultimately looking at both synaptic signaling in the brain, and then as the adult animals beautifully replicated what we see in vivo with dad stress. These animals are hypo stress responsive. So ultimately, what does this tell us? This is not a model of schizophrenia. This is not a model of autism. What this is, though, is an indication that perturbations across the lifespan for mom and dad can contribute likely to the rates of early embryo development that perpetuate into differences in how the brain develops that may give rise to other vulnerabilities when mapped on a G by E by D background. So we're now following up. Morgan Briddy is in the lab doing some fiber photometry, looking at those additional perturbations to these animals by using CRF-CRE and the GC-CAMPS comparison by asking questions of what is it uniquely about these CRF neurons in the hypothalamus that produce these differences in their stress. And so just to wrap up, we have taken these studies translationally. This is a collaboration that we began when I was at Penn, and so was Neil Epperson. Neil is now chair of psychiatry at Colorado, and we're continuing these studies together, looking at humans, because when we began these studies, no one had ever examined the small non-coding RNA composition individually and between men over time. So without knowing what normal looks like, it's very difficult to think about how we might ask questions about men coming home from military service with PTSD who are at reproductive age, or men who have experienced traumas. How do we understand those differences? So this is just a little component of our, this is all published now, you can look this up, that Neil and I have been working on and examining the different types of small non-coding RNA, both the microRNA, the tRNA, and the bottom here are piRNAs across the bottom. This is within subject over time. So these men were recruited from Penn undergrads and medical students. They came in once a month for 6 months. And as you can see, looking at, again, the top here is microRNAs. These are tRNAs. These are piRNAs. We've learned a tremendous amount. We have 25, I think, subjects who completed this study coming in every month. We paid them for their material, and we did a thorough sequencing and a complete modeling and looking at their perceived stress each month with Neil Epperson's group at Penn. Some of the things that came out of this, which were really interesting and never been described before, as you can see, especially in the piRNAs in the bottom, there appears to be a really interesting cyclicity actually to these composition of sperm, which has never been reported before. So what that might mean, we're still working on. And Chris Morgan was able to then model these using our Institute for Genome Sciences and looking at the stress composition to ultimately identify unique microRNA, tRNA fragments, and even a few piRNA that fit with the pattern of perceived stress, whereby time plus 2 months, we begin to see, again, at that stress resolution, changes in these small non-coding RNA that may mean differences for offspring outcome. And this is something we're now doing at the level of zygote microRNA and piRNA injection in mouse zygotes to ask what contribution do they make for development. So I'm just going to end there by summarizing that it's really important, I think, to think about lifetime experiences with trauma as we develop our animal studies and translate them into humans, thinking about the preconception environment, the molecules that happen during pregnancy, level of the placenta, at the level of egg and sperm, as well as I hope that I was able to demonstrate that EVs serve as both a local and a systemic stress signal in circulation and traveling to, in large part, the immune system. But there's been many studies that have demonstrated it's also a key component of brain neurotransmission as well. That stay tuned, I think, is some important biomarkers as well. So just to lastly thank the incredible members of the lab who participated in these studies. I gave them credit throughout. Chris Morgan continues to do the studies as we're working with Neil Epperson on further human studies, including looking at military veterans and changes in sperm pre and post theater exposure, as well as Jennifer Chan, who worked on the animal studies. Of course, our collaborators, including those while we were at Penn, our current members here at the University of Maryland, and incredible thanks to NIH institutes for funding this research, especially NIMH for the R37 that's funded the paternal stress studies. We do a lot of work in the Baltimore community, working on health disparities and racism in the environment, especially with kids in the schools here. So please follow us on Twitter. We're always happy to have that conversation. And thank you.
Wow, thank you so much. That was truly breathtaking. Really great. And now Camille who is one of our trainees, will lead us through some questions.
Great. Can you hear me okay?
Yes.
Great. Well, thank you, Dr. Bale, so much for your presentation. My classmates and I have really enjoyed learning about your work over the course of this semester. So I'll go ahead and start with the first question.
And let's make sure Tracy is still here. I see she's disappeared, at least on my screen.
Yeah, same.
She's exited from the presentation.
Tracy,
like.
Can you hear us?
I think she left. We'll have to get her back.
Okay. I
Amber,
wonder if someone
Amber
behind the scene can help us get Tracy back into the main stage. Let's hold off on asking questions.
Okay.
Hannah just said she's trying to give her a call right now, so hopefully we'll get her.
Okay. Yeah, Laura said they're working on getting her back.
We should remind folks to put their questions into the live Q&A as well, just as a reminder.
Yes. Yeah, so please use the Q&A to write questions, and we'll do our best to take them up. And if we don't get to them here, we can address them in the afternoon question and answer session with all the panelists.
Well, if we can't get her back on, Camille, you can ask Richie the questions, and let's see how he does. I mean, maybe actually what we could do is, Camille, maybe you could just say a few words about your thoughts about the presentation and some of your questions, and we could just even have a discussion about a little bit of some of the stuff as we're waiting.
Sure, yeah. Oh,
can you hear me?
Up there you are.
You're back.
It crashed, literally crashed everything just crashed. I don't know what.
Oh boy.
This platform is still a little shaky, I think.
I mean, it just crashed my entire computer. Anyway, we're back. Sorry.
Good to have you back. So Camille is going to start us off with asking a few questions. These are questions aggregated from the group of trainees.
Great. Go ahead, Camille.
Yes, thank you. Thank you so much for your presentation. I'll jump in. Most prominent models of human stress indicate that stress and its associated psychological and physiological responses result from an individual perceiving themselves to be under threat or stress, rather than the environment or event itself. Is it possible that one mechanism through which epigenetic transmission shifts stress responding is by changing how children attend to and recognize threat in their environment?
Yes. Camille, that was a very long question. Can you give me the punchline for the end there? What are you asking?
Mm-hmm.
Sorry.
the question is it possible that one mechanism, well, I guess it is a yes or no question, but maybe you have more thoughts about it. Possible that one mechanism through which epigenetic transmission shifts stress responding is by changing how children attend to and recognize threat in their environment.
Yeah, absolutely. I think, the field of stress neurobiology is huge, right? We have everybody studying it at all different levels, and it's clear that there is.
I think we just lost her again.
Yeah. Well, one part of the presentation that I really enjoyed was hearing about her work with women in the Black community in Baltimore. That wasn't something that I was aware of based on the readings that we did in our course this semester. And we did develop, my group who read her work developed a question about how might these findings inform interventions for high-risk populations. So if she comes back, I'd like to hear her thoughts on that as well.
The project, the Grady project, has just been a tremendous resource that was, I think, initially started by Carrie Ressler at Emory, and Tanya Jovanovic has maintained a lot of that, and we're seeing the fruits of that work, now and will for years to come.
But Ned, is that the same project that's going on in Baltimore? I thought that was in Atlanta.
Yeah, no, this is Carrie's project in Atlanta at Grady Memorial that he
Right.
started when he was at Emory.
Okay, yeah, but Tracy was also mentioning something in the inner city in Baltimore.
Yes, I think at least some of the data was from the Grady Memorial Project.
Yeah, some of it was. Both that were mentioned. Yeah.
I mean, what's really exciting is that I think that Tracy has opened up an entirely new field. And her imagination, I think, in relation to the questions being asked is really, it's very clear, and also how it's driving some of these questions related to the exosomes and all of that is really interesting.
Yeah, I think that going back and forth between the animal and human is extraordinary, very unusual.
Absolutely. And I think her insights about the timing of some of these events post-stress,
yes.
Is really quite interesting as well, and may have important intervention implications.
Yeah.
Yeah, I mean, maybe what we can do till she gets back is there are questions and comments from I'm not sure if the folks from the audience can actually talk.
Probably not, Richie. I think that's.
Yeah. Okay.
Camille, do you want to just even ask the questions that you were going to ask, and then just to bring up the issues, I think, that we can all hear about what you guys have been thinking about, and maybe we can chime in a bit around that.
Sure, yeah. So the question that she was about to answer was about mechanisms through which epigenetic transmission shifts stress responding, about changing how children attend to and recognize threat in their environment. Another question that we had was Stress is often characterized as a unitary concept, though we are increasingly coming to understand. Is she back? We are increasingly coming to understand it as a multidimensional or multifaceted phenomenon.
Can you hear me?
Yes.
Yes.
Oh my God. Okay.
We're going to ask you another question about stress.
Whatever this platform is, it crashed and.
Oh no.
Oh no.
Poor Tracy.
It's really frustrating for her, yeah.
Okay, Camille, keep going.
Okay. Stress is often characterized as a unitary concept, though we are increasingly coming to understand it as a multidimensional or multifaceted phenomenon. And the question is, do you think different types of stress, for example, deprivation versus threat, incidental versus chronic, are variably transmitted through generations? And if so, why or how? And if so, how much of a concern is it that the stress models in animals do not perfectly parallel human stressors? That was one question.
Yeah, that's a great question.
Should I read more, or did you want to discuss that one?
Read more.
Okay, all right.
There have also been some really good comments and questions from the audience too, which we can just read because they're issues that we can reflect on in the convening this afternoon.
Okay.
But go ahead, Camille.
Okay.
I mean, maybe I'll jump in the interim. Margot asked a question or made a comment, and a question, which I think is really important. She said, first, thank you for the excellent presentation with a strong focus on how structural racism impacts individuals during their lifespan and transgenerationally. And then she asked, do you have any recommendations for building bridges between researchers and diverse communities, especially given the role that the scientific community has played in sustaining marginalization of underrepresented populations? And given the work that you're mentioning, Ned, with the Brady Project in Atlanta, and the work that Tracy has been doing in Baltimore, these are. Whoops, Tracy is back.
Yay.
Maybe.
Let's see how long this lasts.
Let's see how long this lasts. What have I missed?
Well, maybe we should continue on the topic of Margot's question because it overlaps with one of my group's questions anyway.
Okay, so this was a question I was in the middle of reading, Tracy, which is several people endorsed and also has been asked in different ways. After thanking you for your excellent presentation, with a strong focus on how structural racism impacts individuals during their lifespan and transgenerationally. And then the question is, do you have any recommendations for building bridges between researchers and diverse communities, especially given the role that the scientific community has played in sustaining marginalization of underrepresented populations? And I think the work that you've done, Tracy, with both in Atlanta and in Baltimore really serves as an extraordinary exemplar of how these bridges can be built, albeit I think they're still relatively rare. So any helpful insights you can provide would be great.
Yeah, this is a great question. And it was as part of my recruitment from Penn to Maryland was to start this center on epigenetic research in child health and brain development. And part of it was, well, on campus, can we build better translational research? So getting animal studies going up to the level of humans, and then human input from clinicians helping us build the animals. And it became clear that in the city of Baltimore, if we were going to think about recruitment in a majority Black city, of which has tons of health disparities, all kinds of redlining, and just history of abuse, neglect, and marginalization, it wasn't going to be simple. So I spent a lot of time talking with both Carrie Ressler and Tanya Jovanovich about what lessons they'd learned in Atlanta on the Grady Trauma Project, but also working in the community here. So I do a lot of work at the level of Baltimore City schools, because if you work with the kids, then you can start to have conversations with families and communities around those schools. I do a lot of work with city council, who set a lot of policy for the city of Baltimore. So I try to approach it from multiple angles of working as an engagement and continuing conversation to build relationships. I try to stay away from the word outreach, which many in the community can perceive of as being sort of insulting. So I try to not use the word outreach, rather it's engagement, and do a lot of listening. When you come into, as an example, a school district, and you have all kinds of ideas, I want to, interact with these kids. I want them, to teach them science and the beauty of the brain and all. And then I'm going to work with their families. And you come in with the idea that you're going to offer them all of this. And then you quickly realize that that's not how they're going to view it, that individual communities come with their own perspective. And so it really is a relationship building process, that you need to understand their experience and perceptions as much as they need to hear what it is that you do and the value to the research that they're included in it, right? But it's a very sensitive subject, right? This is Baltimore, right? Henrietta Lacks. You need to know no more than that to even appreciate. And so this is become very intergenerational in the community here, to really not trust the process. And so it does take time. It does not happen overnight. And it really becomes a lot of listening and a lot of engagement and learning. I have learned so much. We have a program that we do with the Baltimore City schools with an artist, and we do science, and we did this giant mural on the outside of one of the schools. And this is our program that we do over the course of the year. And every kid participates, the families, but it took that entire school year of continuing, having family night, having like, what are you doing with my kid? What's going, and there's a lot of distrust, and it's been earned in this community. So it just takes time. And I think you can't do a one-off, and you can't just come in and think that everybody's going to embrace you, because they're not. So it does take time, and I continue to learn all the time. The city council has been a great experience learning. They've passed the city of Baltimore, the Trauma Informed Care Act, and they're now taking it to the state to pass it as a state law. It informs everything from education to incarceration to medical care, how trauma, something we're all interested in, and needs to inform care, and that it costs more to educate children who've experienced these high levels of trauma than it does kids who may, be out in the suburbs. Mm-hmm. So I spent a lot of time educating city council, so that they now use words. It's great. On Twitter, I see our city councilman using words like brain, neuroscience. So it's a process that takes a lot of time.
That's great. Thank you.
Absolutely.
So, Camille, do you want to ask another question, or we can go to the questions from the group? I don't know.
How about if there are more questions from the group, I feel like there was supposed to be time for other folks as well. So.
Well, we can take one more question, I think. This is a question from Carly. Concerning the studies in the trauma survivors in Atlanta, did you assess when the participants underwent pubertal onset? Building on that, can you elaborate about the decision to bin the ages at less than 14 and 14 to 17?
It's a great question. So these were retrospective. So these were women who were already in the Grady trauma database. And so we took them, a large group of women, and there's like 120, and we assessed them all with high level of trauma, matched them for PTSD symptoms, and then looked at them from the perspective of a unique, specific type of traumatic experience, interpersonal violence, that of sexual trauma, and then divided them by a typical onset in terms of, brain maturation of adolescence being qualified as ranging-ish 14 to 17. So we're not specifying based on puberty. We're sort of defining it as social determinant of adolescence. There's a lot of different ways we could bin this. There was we have no data to collection of when women went through puberty. So because it's retrospective, we don't have that data, unfortunately. So this was more based on social determinants of sort of an adolescent window versus earlier than that versus adulthood.
Great. Thank you. We're a little over time, but there's one additional question I'd like to ask. We can just talk about this briefly and maybe talk about it more later. As we know, there's a lot of variability in how humans respond to stressful and traumatic events, and there are some who show patterns that we consider to be resilient. And I wonder if you've thought about looking at using the same beautiful, elegant, mechanistic approach you've been using to look at the transgenerational mechanisms of trauma to see if there are transgenerational expressions of resilience.
So Richie, in the animal studies?
Yeah.
Yeah. We've thought a lot about this, both from the perspective of these marks surely can't be permanent. So there must be some way in which the environment might either reverse them or produce a different signal. But we really struggle because mice are just so stress reactive as to what sort of a resilience building might be. So, would it be something rewarding? Well, something rewarding could also be caloric.
Yeah.
So we have.
What about some kind of like enriched environment kind of?
Yeah, we go back and forth on this because for mice, one man's enrichment is another man's stress, and so for mice, it kind of all lumps together.
Yeah.
And then if you start doing something like wheels, that's exercise, and so, I mean, to me, they're all sort of just different environments. Because they're all going to produce a stress response in mice. Mice are just particularly stress reactive individuals. But yes, it's a really unique and interesting way to think about it, Richie, that we have talked about. How could we, if evolutionary perspectives are important, then if a stress produces one signal in dad's somatic cells, then shouldn't something that's enriching, such as availability of high-fat food or something that's beneficial to the environment in terms of evolutionary perspective, could that reverse it or produce a different signal? We haven't done it yet, but it's something that we're interested in. Yeah.
Okay.
Yeah, resilience I think is critical to think about as well.
Cool. Okay. Well, I'm sorry you had so much technical challenges. This is the first time we're using this platform. We've all had it today, so you're not unique in having this.
But we'll have a chance to talk more with Tracy for the overall discussion, which will be great too.
Yeah.
Sure. Great.
Tracy, it's fabulous work, and thank you again for your presentation.
Thank you again.
And we much appreciate it.
So we'll resume again at 2:15 Central Time, 3:15 Eastern Time, for the last talk of the day by Josh Gordon. Thank you.